Funding for this podcast is provided in part by the IPM Network through the USDA National Institute of Food and Agriculture and by various commodity groups through checkoff dollars, including the United Soybean Board, National Corn Growers Association, and Cotton Incorporated.
The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center and hosted by Ed Zaworski.
Welcome back to the I See Dead Plants podcast. As always, I'm your host, Ed Zaworski. Today, just like every other podcast, I got a really interesting one here for you folks. I have Dr. Jeri Barak here from the University of Wisconsin-Madison. And we're going to be talking about microbes, but not entirely about plant pathogens. So it's going to be a really fun one. And as always, we're going to give some background information on her. And I'm actually really excited about this. I think I've mentioned it on this podcast before, but I am kind of a fish head, Jeri. So I'm an avid fisherman, but I also breed and raise freshwater fish from all parts of the world pretty much. And so I was really excited to see that you got a bachelor's in marine biology, and I was kind of curious about that, and we'll get into it. But then I went on to get a PhD in plant pathology at UC Davis. She did a stint at the USDA as a research microbiologist and is currently the Associate Dean of Academic Affairs at UW-Madison. So, Jeri, I like to start off with some get-to-know-you questions, and I really want to hear this one because of the marine biology portion, but how did you get into plant pathology, and what caused you to make the switch?
Great question. So I got my degree at Wisconsin State University. Let me back up one.
Yeah.
My first year in college, I was actually at Boston University, and I was started in the marine program, and I realized quickly that I couldn't afford to stay there. And so I met with my advisor at that time, and he suggested that I transfer to San Jose State, where tuition was cheaper. But he said you could come back to the BU Marine program that was hosted down in Woods Hole at the Marine Biological Lab. And it's a semester program. It's taught in block, meaning you took one course for a month. And it was kind of the best of both worlds. So I got the best attributes of the private college marine program, but I mostly paid tuition at the California State program. So when I went to I met Dr. Ivan Valiela, and I joined his lab and started doing research. And after the semester was over, went back to San Jose. But the subsequent summers, I would go back to Woods Hole and work in Ivan's lab. So when the time came for graduate school, I applied for a doctoral program in Ivan's lab, and I was accepted. And started my research. And I was studying eutrophication in an estuary that had fresh water flowing into the estuary. And of course, the estuary was saltwater. And every summer, this estuary was becoming anoxic. And so the question was, what were the drivers for this? And so I started doing some field research. And several people in the lab were working on this. And what the lab figured out was that people that lived on the rivers that fed into the estuary, they were not connected to the city's sewer system. And so the phosphate that was generated in the houses were leaching into the rivers that would go into the estuary. And that was driving the anoxic blooms that happened in the fish gills.
Interesting.
So the recommendations were that each house would have to have secondary sewage system, which were expensive. And so there became a conflict between people that lived in the area and researchers. And the conflict would manifest itself that people would stir up your research side.
Gosh.
So at the same time, it was like one of the coldest winters in 100 years in the cold. I kind of miss Northern California weather. And the idea was that I was probably not going to be able to do my research in this area because of the conflict. And at the same time, I wanted to do, during my doctorate, basic and what applied research. And Ivan was kind of cool. And his opinion was that for a PhD, you should focus on basic research. And so that was a conflict with me. And so I started to search in journals for research that was solving problems and was a little more applied. I came across plant pathology. In the spring, I went and interviewed at UC Davis and Berkeley, and one of the PI was telling me about his research that was done on a farm. And I said, whoa, how'd the farmer take that? And he was like, what do you mean? He's like, not only was he supportive, they fund part of the research. And I was like, wait, you get to answer questions that farmers are interested in and they're not tearing up your stuff? Like you can do basic and applied research and it's not freezing cold. So I quit my first PhD program and I went to UC Davis to become a plant pathologist.
Makes sense. I don't know if serendipitous is the right word to use here, but it's this is the first time I've heard of somebody being in a PhD program where civilians are tearing up their research. But yeah, it seems like a good transition from there because usually when the people are happy to see a plant pathologist. Well, I guess unless they're coming to check out a disease outbreak or. But we're I think that overwhelmingly people know we're looking for solutions to the disease problems though, right? So,
Definitely.
So from there, Jeri, I'm kind of curious, like where did I had to look all this stuff up. So I'll ask for like a clarification from you about a lot of the terminology probably as we go, because coincidentally, my wife works in a hospital and she does like the testing for blood work and all that stuff. So she knew exactly what I was talking about when I said enteric human pathogens. I guess, yeah. Would you mind explaining what an enteric human pathogen is for our listeners and how did you start to study them?
Right. So I was finishing my PhD in 2000 and in the late 90s, we had some of the first outbreaks of gastroenteritis caused by either Salmonella or Shigatoxin E. coli in association with eating plants. So you might remember, well, you're young, you may not remember, but we had the Odwalla outbreak with unpasteurized apple cider, apple juice.
Okay.
There was the spinach outbreak later in 2008. There was a huge multi-state outbreak. And even some young people died of consuming raw spinach that was contaminated with Shigatoxin E. coli. And that's the one that it's more infectious, meaning it takes fewer cells. And some of the outcomes are more negative to humans in the sense you can be put into a coma. It can cause kidney failure, leading to the need for kidney transplants or dialysis. So the Clinton administration funded research to look at these enteric pathogens in association with plants, and the funds went into ARS USDA. So that was right as I was finishing my doctorate. And ironically, I wanted to leave academia because I was convinced everyone in academia was crazy. So I went to work for the federal government. So I did a postdoc for about a year and a half. And then my scientist, the lead of the lab, left her position. I applied for her position. And at that time, there weren't very many people that worked with enteric pathogens in association with plants. And so I became the leader of that lab. I stayed 7 years. Then the position at Wisconsin opened. And by that time, I was absolutely sure that everyone in the federal government was crazy. So I went back to academia.
Jeri, for those who are listening, what separates an enteric human pathogen from any other pathogen?
Good question. So these are bacteria that live inside the human body. So that's 37 degrees Celsius. So most bacterial plant pathogens cannot live at that temperature. Now, some of the cousins of these enteric pathogens, like Erwinia, some strains, some species can live at that temperature.
Gotcha.
But the more common bacteria that we study in plant pathology, like Xanthomonas and most Pseudomonas, do not thrive at that temperature. You have outliers like Pseudomonas aeruginosa, which is both a human pathogen and a plant pathogen. But those are rare. The enterics thrive in mammals, but Salmonella also can thrive in avians.
Oh, yeah.
And reptiles. So Salmonella actually has a wide temperature from 42 degrees Celsius to 20 degrees Celsius.
Gotcha.
E. coli is not that broad.
Gotcha. It's interesting to think, I don't often think about humans, right? I think much more about plants and their diseases, right? It's interesting, I'm sure I've heard it put this way before, but I don't often think of it that way as to why our body temperature is what it is. It makes it inhospitable for a lot of pathogens, so it makes sense. So these enteric pathogens are the ones that can survive and thrive in that temperature range, right? So beyond that, getting to know you a little bit, I want to get into your research. Can you give us, Jeri, just kind of an overview of what your lab currently works on?
Yeah, good question, because turns out I've been doing this for 26 years. So
Perfect.
What we started doing in the beginning is not at all what we do now. I mean, in 2000, what we were looking at is, are there simple solutions to these bacteria being on leaves? Can we just wash them off? Maybe that's the problem. And so we studied adhesins. We studied, what are the mechanisms that the bacteria can colonize these plants. Nowadays, we take a more systems approach, a simple system, where we think about the human pathogen in the field. It comes across plants that also have other bacteria in them. For almost a decade, we also looked in phytophagous insects, because they're common in the field as well. We're looking for multipliers or for common things that happen that could lead to a food safety problem. We're always thinking about giving farmers something to put in their toolbox. When should a farmer think about food safety? When should their concern really be in that direction instead of all the other things they have to think about?
Sure.
Did that answer your question? I mean, I can go in more depth.
Well, yeah, I had a couple of sub questions I was thinking about along the way. Like, what I know, when we're talking about, E. coli or Salmonella, we're not probably talking about field corn, but what types of crops does your lab work with?
So it's going to sound and even though studying these bacteria on leaves, most people are like, I don't eat tomato leaves.
Yeah.
But raw tomatoes and salsa have been a almost annual food that people eat raw that have caused outbreaks because they were contaminated or colonized by Salmonella. And a lot of our research really focused on Salmonella pretty much since I came to UW-Madison for two reasons. One, I had a young child, and Shigatoxin E. coli are so infectious, 5 cells, and young children are super susceptible. So that's kind of a selfish reason.
Sure. Well,
but from the science perspective, Salmonella is amazing. It's a generalist. There's probably almost no environment on the planet that you won't find Salmonella. And that's not necessarily true compared to E. coli. And I know we call them enteric pathogens, but I would posit to you that we call them that, specifically Salmonella, because that's the way we fund this type of research. Like, Salmonella is an amazing environmental bacterium. Its metabolic flexibility is incredible. I already told you about the huge temperature range.
Yeah.
I already told you about so many different hosts.
Right.
We are so fortunate that Salmonella is not carrying those Shiga toxin genes because we'd all be in trouble if it did. Back to your question. Did I answer it?
What crops? So you mentioned tomatoes,
So we worked on tomato because I was very familiar with Xanthomonas that infects tomato. And that pathosystem is really interesting because there are actually 4 species of Xanthomonas that infect tomato. And so that's kind of unusual in plant pathology. They all cause the same disease, but genetically they're very distinct. I also like tomato because it's also infected by Pseudomonas.
Yeah.
Very different bacterium. And that would allow us the flexibility to ask different types of questions in a single host. And it's relevant because of food safety, because I told you about the outbreaks.
Mm-hmm.
Of course, what people really wanted us to look at is lettuce.
Yeah.
And we have done that as well. So those are the 2 crops that we've really focused on in the last decade.
Makes sense. They're probably they got to be among the most common vegetables that are eaten raw, if not the 2 most common, right?
Yes, they're very common, both grown in the U.S. As well as both infected by Xanthomonas.
Oh, yeah.
So that gives us some interesting opportunities to ask questions.
Jeri, I got a question about like on the human pathogen side. You mentioned Salmonella being kind of present in the environment. I'm just kind of curious. You said it's got a wide temperature range that it can persist at, but how does it get introduced to vegetable crops that are being produced by people? And this gets into your research as far as like how they interact with plant pathogen or phytobacteria, right? But before I get into that, I was just kind of curious, like, just how do they get to plants, and then how long can they persist of their own accord just on a plant, kind of?
Great question. So Salmonella is most likely moved into the agricultural system through water. There have been surveys in the United States looking at surface water, and Salmonella is a member of the bacterial community.
Yeah.
They can also be moved into the agriculture environment through animals. Someone told me that in the South, if you go into a big cantaloupe field and you look across the fields, you'll see some movement. There's iguanas sitting on top of the cantaloupe looking around.
Yeah.
Iguanas don't necessarily get sick from Salmonella, but they carry Salmonella. So do birds. So do cattle. E. coli is also carried by cattle as well as some other animals. So you don't necessarily separate these types of farming.
Sure.
So
gotcha.
Yeah, that's basically the way that it moves into agriculture, like produce production.
Yeah, it's funny. I actually did a podcast with a ornithologist. We talked about both how birds could be viewed by farmers as pest control, in that they eat insects that might be causing issues on a crop, but they are also viewed from a food safety standpoint, in that they could transmit some of these enteric human pathogens. So you mentioned with tomatoes that, these things can be on the leaves, but they can also be passed to us via the fruit. Can they just kind of be anywhere on the surface of a plant, or are there optimums as far as this part of the plant is where they thrive, and this part of the plant is maybe just where they happen to be sometimes? Is there anything like that?
Yeah. So Salmonella can survive on leaves. In tomato production, the leaves touch the fruit, and with bacteria, Salmonella is flagellated, and all you need is a film of water, and they can just move. So once they're introduced, if they're introduced like through irrigation overhead, they would end up on the leaves, and they can move to the fruit. But I have to say that, yes, they can survive for long periods of time, but the phytosphere is not a conducive environment. And we could talk about the research that we have that supports that. Because as you might not be surprised. We don't get to do a lot of research in the field.
A lot of greenhouse work.
Yeah, growth chamber.
Yeah.
Even a greenhouse, they don't really want us to put salmonella in the greenhouse.
That's fair.
So giving salmonella opportunities to survive on leaves, they don't do too well on healthy leaves.
Mm.
The population crashes. It's either lack of nutrients, exposure to UV. It's just not a great place for them. So there has to be something else. And that's what we were really interested in is what are the risk factors? What are the things that help salmonella? But to your point about longevity, there's been studies that looked at pastures.
Mm-hmm.
Salmonella can survive for years in the soil, for years, in fallow kind of field. So it's in the environment. It's not uncommon. So the question is, what happens that we have an outbreak? Like something unusual, something that increase the chances of salmonella surviving or replicating. And that's what we've been really curious about in the last few years. And it also gave us the opportunity to study the plant pathogens and what they're doing on leaves, because close examination of that, there's not a lot of data for that.
So sounds like it's safe to say that these human pathogens, they're ever present in the environment, especially with regards to, perhaps certain animals and water. But sounds like they're surviving, but they're not thriving in those places, right? They're
don't have fast replication.
Got it. Well, this seems like a really good transition into what I've been reading about with some of your publications about where do plant pathogens or phytobacteria come into play and what's happening there that we know so far.
Good question. So as I said before, we were interested in possible risk factors, and I thought a lot about Xanthomonas. Oftentimes a farmer, whether it's lettuce or it's tomato, Xanthomonas isn't taking down the whole crop. It's a yield reduction. Maybe that's the difference between farmer breaking even or not. But we don't have great intervention strategies for Xanthomonas in either of these crops, right? There's nothing you can spray. There's some genetics, but in tomato, a lot of that genetics is GM.
Mm-hmm.
And we know that's an issue.
Yep.
So I was curious about whether or not Xanthomonas somehow helps salmonella to stay around longer. And if it did, how does it help it? A simple way to think about it might be that as Xanthomonas infects the plant, there's a release of nutrients, because we know salmonella is not a plant pathogen.
Right.
It can't infect plants. It can't access nutrients the way Xanthomonas can. So maybe it seems Xanthomonas, but we didn't find evidence for that. So then the question is, well, what does Xanthomonas offer? Well, it turns out that when Xanthomonas infects tomato, that as it goes into the stomata and into the apoplast, it changes the apoplast from an air environment to an aqueous environment, which is good for Xanthomonas too.
Yeah, right.
Bacteria love water.
Right.
And salmonella can't enter the apoplast by itself. But once Xanthomonas makes this aqueous environment, we found out that if a drop of water landed on that water-soaked area, that's what you call the leaf area where Xanthomonas is in the apoplast and made a wet environment.
Mm-hmm.
It's a wild bit of physics. Like the drop of water lands, and within seconds, it sucks in. So if that drop of water had salmonella on it, now it's in the apoplast. And the apoplast, the plant is being infected by Xanthomonas, does have more nutrients. It's protected from desiccation. It's a nice place to hang out. And salmonella will replicate. And then when you have a replicating population of salmonella, you're more likely that you're going to get interaction with the fruit.
Gotcha. Jeri, I'll admit my shame in saying that I had to. This happens to me all the time, actually, in just like day-to-day life. Is people know that this probably happens to you as well, or anybody who's told someone that their profession is, I studied plant pathology, then they assume that you know absolutely everything about plants. And so when I was reading these papers, I had to look up what was the apoplast. And I think that my understanding is still very basal. So would you mind explaining to both me and the listeners what is the apoplast and what's happening there?
Yeah, great question. So on leaves, if you look at the leaf surface, you'll see that there's some holes, and those are the stomates. And the primary purpose of the stomates is gas exchange for photosynthesis, but they're also gateways. And so below the stomate, so if you go in like a manhole, there's the substomatal chamber, and the apoplast is among the mesophyll cells of the leaf. And the mesophyll cells are packed. There's air pockets, there's spaces, cavities, if you will. And these cavities are accessed from the leaf surface through the stomata, down through the substomatal chamber, kind of like a cave.
Yeah.
And it's in that space where Xanthomonas and Pseudomonas that can infect leaves, that's where their infection port is. And it's important to know a little bit about the physics of a plant, physics of the leaf. So you know most leaves, if you put a drop of water on them, you can still see the drop of water.
Yeah.
Because leaf surfaces are very waxy. In fact, the wax layer is very thick. And depending on what kind of leaf, there's all kinds of architecture of leaf wax. But the mesophyll cells don't have that. And so even if Xanthomonas or Pseudomonas wanted to use its type 3 secretion system, its hypodermic needle, if you will, to infect a plant cell, it cannot do it on the pavement cells. That's what we call the leaf surface.
Right.
But in that cavity, the mesophyll cells are not protected like that. So those hypodermic needles can punch through. And it's that cavity that we call the apoplast. Most importantly for the plant, that's where the gas exchange happens for photosynthesis. So the plant has to have these cavities. Because you might think, like, that's not very good evolution. Why don't they just get rid of that? And then those plant pathogens can't infect them.
Right.
But then they couldn't do the gas exchange, which they have to do to make food from sunlight.
Right. So I'm just kind of curious along those lines, like, can human pathogens like Salmonella or E. coli, can they get into the apoplast through the stomata normally without plant pathogens, or is it necessary for the plant pathogens to be there for them to do that?
So I have shown that Salmonella doesn't do it outside. And people that study Xanthomonas and Pseudomonas have found the specific mechanisms that those pathogens use to open the stomates. They force the plant to open the stomate. And Salmonella doesn't do that. E. coli doesn't do that.
Gotcha. So under normal operations, those stomates are not opening probably at the correct timings for these pathogens to move in, or because they still have to open at some point, right?
When bacteria are on the leaf surface, and they can close the stomates, but they can't keep them closed forever.
Got it.
They have to open them for gas exchange. But with the desiccating surface and the UV that happens on the leaf surface, they can keep out a lot of things. And so it has to be, a good opportunity for Pseudomonas, for Xanthomonas, to force the plants to reopen those stomates so they can go in.
Got it. So it's a perfect storm type of scenario, more or less.
Yeah. And one thing we also found that this is important is if you had a severe rainstorm, leaves can become waterlogged. The rain can fill the apoplast, if you will. And so Salmonella, the stomates would be open, and so Salmonella could go in.
Yeah.
So that's without the plant pathogen. But what we showed was that it took the plant pathogen for the environment to be conducive to replication for Salmonella. So Salmonella can enter a waterlogged apoplast, but Salmonella only replicates in an infected apoplast that's water-soaked from the pathogen. Now, whether that's because the infected apoplast is the one where you have more nutrients, or you have the the shutting off of the plant defenses, we can't really say.
Just that it takes more than just water for them to enter that apoplast and start to replicate. Got it. So I'm kind of curious.
They
I mean,
can
like,
enter the apoplast, but they just won't thrive.
right. So, well, based on that, I mean, maybe this is a hard question to answer, or maybe we don't know. I mean, like, they're not thriving in absence of the pathogen, maybe, but are they surviving longer in that apoplast, like protected from UV and that type of thing, or
For a short window of time.
got it? Oh yeah, because they probably don't live very long.
So maybe if you were a lettuce farmer,
Yeah.
or a spinach farmer, because spinach is only in the ground for like 40 days, and you had a water clogging rain event, you might not want to harvest right away.
Got it.
But you may not want to harvest those leaves that are waterclogged anyway because of the way that we harvest spinach
Sure.
or baby lettuce.
Sure. So, Jeri, you mentioned like with bacterial diseases in general on most plants, right? There's not management strategies. I was talking with my wife about it the other day. Like, what if there was, like, what if we were spraying, antibiotics all over massive farm fields? The rate of resistance acquisition for all bacterial pathogens would go through the roof. There
are strategies though that could be successful.
Sure. And that's what I wanted to ask you, like, what do people do in tomatoes and lettuce for bacterial disease management? And is there anything that farmers can do with regards to the human pathogens as far as management?
Well, the farmer needs to manage their disease, their plant disease. Beyond that, it's very difficult, I think, for a farmer. I think post-harvest, we do know things that we could do. We have to bring the consumer along is the problem.
Sure.
For instance, after the 2009 spinach outbreak with E. coli 0157H7, the FDA okayed the use of UV irradiation for leaves, like those prepackaged salads, if you will. But it's never been used in the industry. Can't be used for organic. And some might say that consumers aren't science literate enough to understand that leaves that are treated with UV are healthy to eat.
Yeah, that's interesting. I don't. That's a whole other question entirely. I won't even ask you, but I am curious. Like, I'm gonna have to bookmark that one for later for myself. Like, why is UV not acceptable for as far as like commercial treatment? But so what?
Because if you ask average consumers about UV treatments, some might say, well, then the food would be radioactive.
Yeah.
That's what I mean by science literacy. And as far as our organic is concerned, it's not considered natural.
Jeri, is there, like as these crops are exiting a farmer's field and going to market, are there, I mean, I don't know that I'd consider this. Well, I guess I'd consider it management. Is there any kind of like testing or interfacing done at that point to test if these human pathogens are present? Can that even be done? I'm just kind of curious.
So there is a lot of testing that's done. It's company specific, likely proprietary. And I think they do get pulled off the shelves. I will say that a lot of the outbreaks that happen, like with lettuce, are the ready to eat, salad bags.
Yeah.
I don't know off the top of my head, but I don't think there have been very many outbreaks from heads of lettuce, but that's a consumer preference, right?
Right. Well, that's an interesting extra angle too, because I mean, did that come through the water that they washed the lettuce with before they bagged it, or did that? Yeah.
Well, that's one of the issue, or it could be one of the issues, right? It's highly unlikely that if Salmonella was in a lettuce field, that it would be everywhere.
Right.
I mean, that's not what we see with bacteria. That's not what we see with Xanthomonas. It's splotchy, right? But if you harvest all those leaves and then you dump it into a big vat and you triple wash it, now the 10 leaves that had Salmonella or something on it, you just dumped them in a big bathtub with all the other leaves.
Right.
So that water can be tested. And I suspect that it is tested in a lot of cases. And if there was a positive, I'm pretty sure they could pull those products.
So I'm going to try and do a summary as much for myself as much first for my listeners. Make sure that we're all on the same page. So thus far, your lab has done a bunch of work over the last, what'd you say, 26 years,
Yes.
about these human pathogens with regards to vegetable crops. And so, seems like what you guys have learned recently is that plant pathogens can play a role in how much these pathogens are able to replicate on the plant and survive. Sound about right so far?
Yes.
What's the next steps for your lab, Jeri?
In some ways, the next directions actually are veering away from the human pathogens, but we learned something using the human pathogen.
Interesting.
So I've talked a lot about Xanthomonas, but I said that we were interested in the tomato pathosystem because there were, the two big bacterial plant pathogens, Xanthomonas and Pseudomonas.
Mm-hmm.
And for years in our lab, when we infect plants with Pseudomonas, we did not see any benefit to Salmonella, even though with Xanthomonas, there was always a benefit. And we thought that was kind of odd because Pseudomonas also makes an aqueous apoplast.
Mm-hmm.
So
yeah.
That should be good too.
Yeah.
But it's not. So one of the things we're really interested in is what's the difference between the Pseudomonas and the Xanthomonas? And we're doing a comparison between metabolic outcomes once the same host is infected with either Pseudomonas or Xanthomonas. We don't think we'll find the answer there per se, because Salmonella has such metabolic flexibility that it would likely thrive in either type of infection. But just as a plant pathologist, we don't really know the change to the host that's infected with Xanthomonas versus Pseudomonas. So we're very interested in that. And we've also come across that it looks like Pseudomonas is hostile to other bacteria that come into its infection port. And we're very interested in that mechanism because it's not Salmonella specific. And so that's the direction that we're going, is more fundamental phytobacteriology.
The microbiomes that are happening, well, pretty much anywhere, but I mean, on a plant surface, I'll keep it there, are so interesting. I was thinking of a question earlier and I almost forgot to ask it, but that just reminded me right now. It sounds like at least Salmonella, interacts positively with Xanthomonas, and maybe not so positively with Pseudomonas. Are there any other interactions that these human pathogens have with any other types of pathogens? Perhaps it's not been looked at yet, or whatever you want to say. But are there any fungi or nematodes or anything else that might enable these things, or do we just not know yet?
We don't know a lot. There has been some research with fungi. There has been some research with nematodes. And then we spent maybe a decade looking at Phytophagous insects. And what we found was that common Phytophagous insects, like leafhoppers, aphids, can distribute Salmonella from one plant to another plant. The feeding of these insects increases the survival of Salmonella on leaf surface. So we thought that was important because, like I said, not only should farmers, if they have the infection from the bacterial phytopathogens, they should think maybe they could have a food safety risk, but also from the Phytophagous insects, because the damage caused by some Phytophagous insects may not be obvious to the consumer. We're not talking about a giant caterpillar going and eating leaves, and then of course you wouldn't eat that leaf. But we always wanted to help the farmer and maybe the consumer, because that is one thing that we would say to consumers: you should buy the healthiest produce that you can.
Yeah, absolutely. Well, I'd say, based on what I've learned in this podcast so far, this information is just as valuable to growers of these crops as it is to, or just as valuable to a consumer as it is to the growers. Jeri, at this point, I kind of want to transition into the wrap-up questions at the end here, but I like to pause at this point and kind of ask people, is there anything that we didn't cover or anything you thought you think might be worth mentioning to my listeners about enteric human pathogens or phytobacteria that you've been looking at, or anything that we've missed?
Nothing comes to mind necessarily.
Sure, that's fine. So let's jump right into it. I was kind of curious, there's a lot of talk. I actually was just listening to a podcast this morning about feral hogs and the effects of them moving around the country based on climate change. So, I mean, this is no different. I'm sure that there's some avenue that's going on with these human pathogens as far as them infecting vegetables and, increased rainfall events or increased severe weather events, or you could probably go a different avenue, a bunch of different avenues there. What do you think is going to happen with these pathogens in the future? Do you think we'll see like an increased prevalence or increased range where we see them, or I don't know. What do you think might happen?
Well, we know that those types of pigs you're talking about, that they can carry the shigatoxins and E. coli's. So
interesting.
So they could be introduced into a lettuce field. And what we don't want to happen is like a scorched earth policy, right? You were talking about birds.
Mm-hmm.
We have these pigs.
Iguanas in a cantaloupe field.
Yeah, exactly. Like you don't want to remove all of that from our environment because historically that doesn't seem to work out very well. We cause more problems than we solve. But what we might have to consider is going back to the post-harvest UV treatment. That's a viable option, and we could make salads safer.
Building off of that, Jeri, do you think that what do you think it takes to get to that point where I hear UV sterilization of salad and I'm like, great, let's do it. Less people getting sick. What do you think it takes? Do you think it takes more education? What do you think it takes to get people comfortable with something like that?
That's a really good question. It's slightly hard for me because I don't know what people's reason for not accepting it. There's been good studies that show that the nutrients in the UV treated produce isn't different. I think we're back in an anti-science type of culture that exists right now, and it's just very difficult.
Yeah, that's part of the reason
because you can show people all the studies that the produce is just as nutritious, and people will come down and say, well, I just want the stuff natural.
Yeah.
Well, I guess Salmonella is natural. Shigatoxin E. coli is natural, but it's not good for the compromised.
Sure.
It's not good for the young or the elderly.
Absolutely.
Death is natural too, but I don't think you should support that.
Right. Well, that's part of the reason why I do this as well, is that I hope I can get some people's ears on, a bunch of things. So, Jeri, my next question for you is, if you were talking to a grower who's producing, let's just say, lettuce or tomatoes, what kind of advice do you have for them with regards to, I guess I was going to say with regards to the human pathogen side, but I mean, what advice would you have for them, period, with regards to the human pathogen, the plant pathogens?
I mean, should we have the opportunity in the future to have genetics for tomatoes and for lettuce that reduce plant disease? That would likely have a positive outcome for food safety. Any type of strategies that can reduce plant disease is going to be good, I think.
Sure. Yeah. What's good for the plant in regards to plant disease is good for the people eating the plant, right?
Absolutely. I think the cost structure is the problem, right? The cost structure, because maybe the farmers can't afford it. If it was genetics, the host, getting a better price for farmers. I don't know how we do that. But
sure.
As I understand it, we don't pay that much for food when we think of our total budget. And I know that's hard to say these days because people are talking a lot about how groceries are so much more expensive. But as a proportion of our total family budgets, we don't spend that much for food. And only a fraction of that actually makes it out to produce producers. That's not the type of agriculture that's supported.
That's kind of a bridge into my last question, Jeri. So I like to ask, I like to kind of wrap up these podcasts with kind of a, like a public facing answer. Like I like to ask, like, yeah, if you had to explain to a member of the public why your research is important, what would you say to them? But I was kind of thinking for this one, like more rather than just a member of the public, which it still would be a member of the public, but what would you want a consumer of these products, like you're saying, again, lettuce, tomato, whatever vegetable you want to say, what would you want them to know about this research?
One of the courses that I've teach is about global food security. And one of the biggest drivers is both food waste and production loss. And so this research is important because focusing on solving yield loss due to disease increases, the income for farmers. Because most tomatoes aren't grown in fat economy countries. They're grown in lean economy countries, and they're cash crops.
Mm-hmm.
And the success of those farmers leads to the success of their communities and their families. And so this type of research is important for food security stability globally, in my opinion.
I couldn't agree more. Jeri, this has been wonderful. I have learned a lot, and I'm sure that my listeners have as well. At this point, I like to leave time at the end for you to make any acknowledgments that you would want to make, point people to any resources, point people to any research you've done. You've got the floor.
Oh, thank you. Well, the great thing about being a scientist, being a researcher, is the team that I get to work with. Because believe it or not, you spend all your time learning in school, getting the doctorate degree, getting the research job, becoming a professor, and then you don't get to do the science yourself. So everything that I've talked about is due to the students I've had, the undergraduate students, my graduate students, the scientists that has been with me like almost my entire career at UW-Madison. It's a team effort. And it's actually what makes it a lot of fun. The collaborators I've worked with for years. Turns out we don't grow very many tomatoes in Wisconsin. So definitely a shout out to my collaborators at University of Florida in Gainesville, that they let me come down and do some work, sometimes outside without salmonella. And I've just been so fortunate to work with some of the best and getting up every morning, wondering what happened in the lab is what gets me out of bed. So plant pathology gave that to me.
I got one more question for you, Jeri, to close it out. I'm just kind of curious now. You mentioned at one point in your career, you thought that everybody in academia was crazy and then everybody in USDA was crazy. What do you think now?
Yeah, good question. Actually, I love being a professor, being around the students, the energy that they bring to the science, the crazy questions they ask you that you've never thought about, their interest in where they want to go with their research. There's no year that's the same. There's no month that's the same, no decade that's been the same. And science is just this living entity. And I've been so fortunate to be a small part of it.
Wonderful.
And it was more the systems that made me crazy. I loved my colleagues at the USDA ARS. I love the training that I got at UC Davis, the training that I got in Woods Hole. All of it has brought me to where I am today. It's the system that's challenging.
I couldn't agree more. I do think that we are all a little crazy, though, but I wouldn't have it any other way. So with that, folks, I hope you enjoyed this conversation with Jeri. Thanks for listening, and we'll see you next time.
Thanks, Ed.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ntipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Music written and recorded by fellow plant pathologist Sean Toporek. Thank you for listening to I See Dead Plants.